Alright, welcome back everyone. Um, Today we're going to be talking about chapter 10. It's about photosynthesis. That's the process that involves plants, as you may know by now. Um, it's a process that's required for all living things to function, uh, in I guess, the normal way. Organisms, animals and humans and so on, they need plants to perform photosynthesis to accomplish, the life cycle, right? To be able to maintain themselves. So photosynthesis is basically the way that plants capture light energy, right? And also uses carbon dioxide from the air and produces oxygen, glucose to maintain itself, right? And water as well. Uh, so let's see here. So let's begin with this. This is just a picture of a tree. You know that's one of the examples that performs photosynthesis. We're gonna try to also run this kind of fast, this chapter, because it does contain a lot of information, a lot of slides. So just pay attention, and if you need to like slow things down and repeat it, please feel free to do so, and make sure you're following with me in the book, in the PowerPoint. Because again, some things you need to basically pay attention to close detail. If I slow down a lot, it'll take me maybe 4 hours to do the PowerPoint. So I'm gonna explain it kind of in a fast way, and make sure you watch the animation videos on the PowerPoint. Um, they help a lot. Like visuals in science go a long way. So make sure you see those videos so you can get an idea and perform like a little story and a little like visuals in your head of how those molecules and how things actually work the correct way. Okay, better than just seeing it in front of you on this on a slide or in the book. Okay, so how do photosynthetic cells, right, use that light, right? So there's a certain way that they do that to produce the organic molecules like sugar and oxygen, okay? So this is what we're going to talk about today. So this is a life cycle, as you can see here. You have like the plants use the photosynthesis to capture light energy from the sun, and they use their chloroplasts. Do you remember that organelle? Um, that's the main one that basically is responsible, and this is shown right here. The whole thing is a chloroplast, right? And it's got those little green stacks, and they're called thylakoids. And so this is the main one that's responsible for all the photosynthetic steps in order to capture the light energy and produce your organic molecules and oxygen at the end, okay? Uh, and so, other organisms like animals and like humans, right? They consume those plants, right? And they basically use cellular respiration to give off, to create first ATP, right? As we learned from the last chapter, and also they give off heat as a byproduct. And in the process, cellular respiration creates carbon dioxide, which is exactly the opposite of photosynthesis. Carbon dioxide in water, okay? So photosynthesis, just the definition, is the process that converts solar energy, right, light energy, into the chemical energy, which is like, organic molecules, sugars, things like that, inside those chloroplasts, okay? Um, Again, photosynthesis is, required for the entire living world, right, directly or indirectly. We will see, like, how is that possible. Okay, so autotrophs, when we say autotrophs, think about self-feeders. So auto, like anything else, like when you say autograph, for example, you're talking about your own self, okay? So that would be self-feeders. Remember, auto is self-feeders. And an example of that would be any plants that perform photosynthesis, which are all plants, right? These organisms, right, like plants, they don't rely on other organisms to obtain their food, right, and their nutrition. They actually created themselves. They do so through the process of photosynthesis. Okay, so autotrophs are producers. You see that? That's what they produce things, right? They're not consumers, but producers. Okay, they produce your organic molecules, right, using harvested carbon dioxide, right, from the air. That mainly comes from what it's already in the air, but it gets increased from the other organisms like animals, humans, industrial stuff, right? Everything that creates that huge amounts of carbon dioxide in the air, those plants are basically benefiting from this. Okay, so again, all plants are photoautotrophs, so that means they're using their photo is like, talking about like light and sunlight specifically. An autotroph, they're making their own energy from that sunlight in the form of organic molecules. So light converted to chemical energy. Okay. Now, um, photosynthesis can occur in algae, right, and certain other protists and some prokaryotes, some bacteria, and so on. They do perform photosynthesis, as we'll see, some examples, but they're a little bit different. They don't have like basically all the steps. They have some steps of photosynthesis just to maintain themselves. Okay, we will see what these steps are. All right, so these are just examples of these photo autotrophs, okay, that we talked about. So they use the light energy, right, to convert it to organic chemical energy. Okay. Oh, and this is the prokaryote example, like we were talking about. So it's just a bacteria, and it uses like um, produces sulfur as a result. So this is and cyanobacteria is another type of bacteria. So these are some prokaryotes that perform photosynthesis. Okay. So when we talk about heterotrophs now, instead of autotrophs, we talk about heterotrophs. These are basically the ones that consume the plants. Okay, so other organisms like animals and humans, right? They're consumers. You see that? So they eat those plants basically. Um, so there are some that are called decomposers. So they consume dead organic material or feces. So they basically decompose. So it comes from the word basically. So um, anytime like for example fungi is anytime you see a fungi, that's a decomposer, right? It basically decomposes things uh, into like its simplest organic and inorganic material. Okay. Almost basically all heterotrophs, including humans, right, depend on the photoautotrophs for food and oxygen. So we need the plants to perform what they do, like photosynthesis, to maintain our cells. Okay. I mean, even eating, even if you don't eat the plants, right, and vegetables and so on, even eating like just completely animals, right, animal food, whatever, and dairy products and things like that, right? These come from animals, correct? And some animals eat other animals, but a lot of them actually, like cows, for example, they feed on greens, right, and vegetables and things like that. So that's why if the greens are not there, they're not gonna be able to live. So you can see how this whole thing is related with each other. Okay. So Earth's supply of fossil fuel was formed from the remains of organisms, right? So organisms that were like decomposed, um, that died hundreds of millions of years ago. They represent the stores of the sun's energy. Okay, the sun that was captured by these organisms, it's basically stored inside them for many millions of years. Okay, so fossil fuels are being consumed faster than they are being replenished, right? Nowadays, okay, due to the many uses of fossil fuels in our daily lives. Okay, researchers are exploring methods, right, of using photosynthetic process to produce alternative fuels. Okay, so there's always like ways that scientists are going about to replace and create new way of energy. Okay. Okay, so let's talk about now we're in 10.2. So photosynthesis converts light energy to the chemical energy of food. Okay, as we said. So light, it converts it into glucose or organic molecules. Okay, so they do that using their chloroplast. Here's a depiction of that chloroplast. Okay, inside the cells. Um, Basically, they have those green structures we call thylakoids, right? Um, and around the green structures, we have a fluid filled, uh, you know, uh, inside that chloroplast. So that fluid is actually called stroma. See that? Okay, so a stack of these thylakoids, we call them granum. Okay, so stack of them are granum. Stroma is the fluid that is around um, the thylakoids, and we will see even the location is actually important because photosynthesis has several steps, and some steps are actually taking place in the thylakoid membranes, and some other steps, okay, are actually taking place in the stroma, kind of like what happened in mitochondria. Okay, in mitochondria, do you remember like some things take place in the matrix of the mitochondria, some other things, some processes take place in the membrane, in the inner membrane of the mitochondria. So this is kind of similar. Okay, that's why actually mitochondria and or chloroplasts are, are extremely like similar in a way, but they consume and produce energy a little bit differently. Okay. Okay, so these chloroplasts, scientists have discovered that they most likely were evolved from photosynthetic bacteria initially, okay, like many thousands and millions of years ago, okay. Okay, so regarding the plants, right now, like where do plants actually have the most chloroplasts? This is where the most okay, so let's continue. So the uh, the location of photosynthesis take place, mainly in the leaves of plants. Okay, I know plants are made out of the roots and trunks and many different parts, but mainly the leaves are the location of these photosynthesis processes, and this is why they're green. We will see how their green color is actually important for that process. Okay. So chloroplasts are found mainly in the cells of mesophyll. So mesophyll are cells inside the leaves, and we will see basically a depiction in a bit. This is where chloroplasts are found mainly. Okay, they're inside that leaf. So veins, as you can see here, right, in between the mesophyll cells, transport water from the roots and export sugar, right, to non-photosynthetic parts of the plant. So whatever is being produced here in the leaves, it gets shuttled through the veins, and that goes all the way to the roots and to the trunk and so on. Okay. So CO2 that the plant actually needs, right? CO2 is a reactant. Okay. So it enters and O2 exits as part of photosynthesis, right? We will see how that happens in a bit. You know, exits the leaf through microscopic pores. So these leaves have actually very small pores that you can't even see with the naked eye. And these pores are called stomata. Okay? So the chloroplast has an envelope of two membranes, right? Similar to mitochondria, right? Surrounding a dense fluid called the stroma. Do you remember that? That's the fluid that I said. It's around those thylakoids. Okay. Thylakoids are these green discs-like, right, inside the chloroplast. They're stacked on top of each other. Um, that some of the photosynthesis steps happen in it. Okay. And they're composed of a third membrane system. Okay, so they're membranous, but they're shaped like discs. Okay, so the stack of thylakoid, we said it's called grana or granum. Okay, chlorophyll. Basically, when we say chlorophyll, we're talking about the pigment. Okay, that gives leaves their green color. Here you go. Resides in the thylakoid membranes. Okay, so you have the thylakoid, which is a disc-like, right? And they're stacked on top of each other. Inside of that thylakoid, okay, you have those pigments, we call many of them actually, and you will see how they're basically very important, probably the most important part. Okay, and these pigments, the chlorophylls, they have a green color, and we will see why. Okay, and what their job is. Okay, so looking at a cross section of a leaf. Okay, so here's a leaf, and looking at it, like if we zoom, okay, so cross section, we're looking at what? These green cells are what we call mesophils. Okay, and here is a close look at one of these mesophyll cell, and you can see that one of these cells have many and hundreds of chloroplasts. Do you see that? Okay. So inside the chloroplast is where we find these thylakoids. Okay, so you can see how microscopic these things are, and at that microscopic level is where these photosynthesis processes are happening. Okay, now remember these pores that we talked about? They're called stomata pores. These are located on the leaves and allows things to go in and out of that leaf, as you can see here. So CO2 comes in and O2 comes out. Okay. Okay. So again, you guys can watch these videos to help you understand. Okay. I'm gonna keep going. So photosynthesis is actually not just one step. It's a complex of series of reactions, right? That eventually produces these three things that you see here. So it produces the organic molecule, right, which is in the form of sugar mainly, okay, that helps plants maintain themselves, okay, and helps conserve the energy. They produce oxygen, which is important for, right, other organisms like us, and they produce water in the process, okay? Excuse me. So the overall chemical change, right, during photosynthesis is the reverse of cellular respiration. So if you remember from last chapter, cellular respiration is exactly the opposite. It basically breaks down organic molecules like sugar and glucose, right? Do you remember that? And it uses oxygen in the process, right? Why does it use oxygen? Because it uses oxygen as a final electron acceptor. Do you remember that from last chapter? Okay. and in the process CO2, water, and heat is produced. Okay, so exactly the reverse of photosynthesis. Okay, so let's talk about basically the reactants, okay, of photosynthesis. So we said here that photosynthesis have the reactants, which are on the left side of an equation, right? They're CO2, carbon dioxide, water, and light energy. So we're going to talk about how does CO2 and water react with each other, okay, and how do they conserve their um, different atoms, okay, and how they form these sugars, and how does O2 come about. So chloroplasts, so those small organelles, okay, that we're talking about, they split H2O, and we will see in detail how this actually happens, okay, and why it's very important. Okay, so they split H2O into hydrogen and oxygen, and as you can see here, so oxygen, hydrogens, do you see that? Okay, incorporating the electrons of hydrogen, okay, into sugar molecules and releasing oxygen as a byproduct. So we will see how this oxygen here as a byproduct is being released, right? This is what we see here. I'm just trying to show you back and forth just to help you follow up on where we are in the steps, okay? So O2 is the byproduct, okay? And we will see how the electrons that are here in this H2O are actually used along with CO2 to create these other products, okay? Okay, so photosynthesis basically reverses the direction of electron flow compared to respiration, okay? So it's exactly the opposite. So do you remember from respiration, O2 is the final electron acceptor. Do you remember that? Now in photosynthesis, we're going the other way around, right? We're producing O2 instead, okay? And in the process, we're using the electrons harvested from water, and we'll see how that happens in a bit, to drive the process of photosynthesis forward, okay? So as we will see that photosynthesis is a redox process. So when we say redox process, we're talking about some reactants are being oxidized, as we said before, and other reactants are being um, reduced. Okay, so the electrons are basically going from one reactant to another. So one is basically losing electrons, one is gaining electrons. Okay, so in this case, H2O is oxidized. Okay, so H2O, when it gets split in the chloroplast, right, it loses those electrons, as we said. Okay, who gains those electrons? The CO2. So CO2, we will say there it's reduced. Okay, so it's reduced to what exactly? It's reduced to its what, organic molecule, right? So that would be the glucose. Okay, H2O is oxidized to O2, we say, or oxygen. Okay, oxygen gas, which is released in the air. Alright, photosynthesis is an endergonic process. So it's exactly the opposite of cellular respiration. So cellular respiration was exergonic. Remember, it releases heat at the end, and it breaks down complex molecules like a glucose, right, into smaller molecules. Okay, and that will be an exergonic process. Endergonic process, that means we're basically um, uh, producing energy. Okay, we're producing and harvesting organic molecules from um, smaller molecules. So CO2 and in this case CO2 and water are becoming bigger molecules like sugar or the organic molecules, right? And the oxygen gas and water as well, okay? Yeah, sorry, the water is actually inside. I got a little bit mixed here. So the water is for cellular respiration, sorry. For photosynthesis, we only have the water being a reactant, okay? So it's consumed. Okay, so this is when we talk about redox reaction. So CO2 is becoming reduced, as we said, right, to your sugar, organic molecules. The H2O is becoming oxidized to this O2 gas oxygen, okay? So, there are two main, let's talk about the two stages of photosynthesis. So there are two main stages. One is called light reactions and one is called Calvin cycle. Okay, so right off the bat, in the light reactions, this is where the plants use light from the sun, okay, to start creating the products. So the first product that's created from the light reaction is your O2, okay, so the gas, okay. We will dive into them a little by little. So the Calvin cycle is where the CO2 or carbon dioxide comes into play. So in the Calvin cycle, we will see how CO2 as a reactant is used up to produce your organic molecule or sugar at the end. So just so you can see where we are, okay? I'm trying to basically organize our thinking of where this is, okay? Because it's gonna get a little bit more complex. Okay, so let's talk about the light reactions that happen where? In the thylakoids. Light reactions happen in the thylakoids. Okay, we will see that Calvin cycle actually happens in the stroma. Okay, so remember this point. This is where studying comes in. So, what happened in the light reaction? We have water. It's split. Do you remember that from a few slides ago? We said that water is split and is actually what? Oxidized. As a result of that, we have a release of O2 oxygen as a byproduct. Okay, hopefully this is making sense. And the whole reason why we want this to happen is to harvest the electrons there are between the oxygen and the hydrogen atoms. Okay, so so this whole process, it reduces the electron acceptor. Here we go. Do you remember that? The NAD. But this, in the plants, it's called actually a little bit different. So it was called NAD plus, right, in the cellular respiration. Here it's called NADP plus. Okay, so don't get this confused. This is the oxidized form because you can see a plus here. Okay, so this molecule is what accepts, so anything that's oxidized can accept electrons from what? From the splitting of water. Here we go. From the splitting of water, those electrons come here and this becomes reduced to NADPH. Okay? And ATP is formed. We will see how that happens. It's very similar to cellular respiration and the electron chain. Do you remember? Electron transport chain? How ATP is produced, okay? So in this case, we say that ATP is basically generated from ADP, remember, adenosine diphosphate, by a process called photophosphorylation, okay? So this is the whole, I know it's complicated, I know it looks complicated, right? But this is what, what produces ATP, okay? Alright, so the Calvin cycle, which happened in the stroma, do you remember that? It forms sugar from CO2 using ATP that was produced in the light reaction, right? We said ATP is generated here, okay? So Calvin cycle uses that harvested energy, okay, and it produces sugars from CO2. So we're making a more complex molecule, okay? It uses ATP and NADPH that was produced here. So these two are being used up in the Calvin cycle. Okay, so it begins with carbon fixation. So we have this CO2, okay, and we want to fix this carbon from CO2, okay, into an organic molecule that's already is in the stroma, okay. We will see what this molecule is in a bit when we talk about Calvin cycle, which is a little bit later, okay. Then it reduces that fixed carbon, right? So we want to reduce this to what? To carbohydrate molecule or sugar, okay? We do that by transferring electrons from NADPH. So this is how we use that electrons that we stored in this, right? From here and from the water, right? We use it in the Calvin cycle to produce that carbohydrate. So hopefully this is making all sense now a little bit. Okay, if you need to repeat these things, you feel free to do so. Okay, chloroplasts basically use that light energy, right, to make sugar, as we mentioned, by coordinating two stages of photosynthesis. This is just a summary for you. Okay, so we have the light reactions, and we have the Calvin cycle, two stages. Okay, light reactions, we said, occur in the thylakoids, right, while the Calvin cycle occurs in the stroma. Light reactions that occur in the thylakoids release NADPH, right, from the water, and at the same time produces ATP, that both are needed for the Calvin cycle. So that means if we don't have these, can we actually proceed to the Calvin cycle? No. Okay, we do need the electrons that are stored here. We do need the energy that's stored in ATP to be able to actually make, right? Because we're going from a small molecule of CO2, we're making a bigger molecule of carbohydrate, right? Or sugar. So that means we need energy, we need electrons to be able to create that. Okay, so let's start talking about the light reactions in a bit. So you are looking at the chloroplast here, that's the organelle that's responsible for photosynthesis. These, the green stacks, are thylakoids. Each one is thylakoid, okay? So this is telling you what's going on. Hopefully this is making sense, guys. I know it's hard. It's not like easy stuff, and it's new stuff too. So just keep focusing here, and you'll get the hang of it. So we're using in the light reactions, it's called light, so that means we're using light or sun energy, okay? And we're using H2O, remember that? Okay, CO2 has nothing to do here yet. Okay, so CO2 does not start until your Calvin cycle process. Okay, so we use light and H2O together. We will see how the light is used in a bit. Okay, it's not shown here. Okay, alright, so we use that along with NAD plus, P plus, sorry, that's already inside the thylakoid. Okay, and we use ADP, okay, that's already in here as well, to produce the reduced forms, which are NADPH, right, from that gains electrons from H2O, and we produce ATP as a result. Okay, here we go. This is showing exactly what we just said. In the process, we have the O2 in the gas form is produced. This is where that O2 comes from when you see the photosynthesis reaction equation, okay? Okay, now looking at the Kelvin cycle, this is just a summary of the whole thing. Kelvin cycle is where, so C for CO2. CO2 comes in, combines with, it uses we fix the carbon from that CO2 on a already available molecule, okay, organic molecule here. It's called oxaloacetate, by the way, so uh, you've seen that molecule before, but anyways, so it gets mixed up here, okay. To do that, we're gonna use what? ATP that was harvested here, and the NADPH that's carrying those electrons from the light reactions to be able to produce that bigger sugar molecule, okay. Here you go. Hopefully this is making sense. Okay, go ahead and watch this on your own. Let's see here. Oops. Okay. So light reactions convert solar energy to chemical energy of ATP and ADPH. Okay. So as you can see, the chloroplasts are solar powered chemical factories. So they're using the solar power from the sun to create those sugars and carbohydrates. And their thylakoids, right, transform light energy, as we saw in the light reactions, into chemical energy of ATP and ADPH. This is just a summary also. Okay, so let's talk about just light in general, okay, and what it's composed of, and how it's actually how this composition helps drive photosynthesis. Okay, so we say that light is electromagnetic energy. It's also called electromagnetic radiation. Okay, so just remember these things. Okay, electromagnetic energy travels in rhythmic waves. So there are waves. Okay, so when we say waves, I'm sure you've seen like a wave of sound. Okay, so wave of light is similar. Okay, we have different wavelengths, and we have a distance between each wave, right? Each bump in the wave, we call this wavelength, right? In between the crest of electromagnetic waves. Okay. Again, they range in how long their wavelengths are. Electromagnetic spectrum, right, is the entire range of electromagnetic energy or radiation. Okay. The visible light, we will see all of these things in a bit. So the visible light consists between the 380 nanometer to 750 nanometer. So we're talking about visible light, meaning that the white light, is divided into the different colors that your eyes can actually see. So it starts from the purple or violet color, which is basically have the shortest wavelength, and then it ends in the red color, which is the other side, okay, of that visible light spectrum. Okay, you see that? So these are the different spectrum of electromagnetic wavelengths. Okay, so we can have gamma rays, X-rays, UV light, infrared, so on. Okay, and you can see how they range in the wavelengths. So we start from this side is very short, 10 to the negative 5 nanometers. So you can see how short that thing is. Okay, and goes all the way to 10 to the third meter in this case. Okay. Now talking about visible light actually falls in this little white section only. Okay. Now white light is divided into the different colors, as I mentioned. So it starts from purple and it goes all the way to the red color. Purple is you have your 380 wavelength nanometer. Okay, that's got the most energy in it. Okay, and then the red has got your lower energy, but because the wavelength is the longest in this case. Okay, So light energy, it behaves as though it consists of discrete particles. We call these particles photons. Okay, each photon has a fixed quantity of energy, so these have energy. Okay, and this is mainly what happened in the sunlight and how this energy actually excites electrons. Okay, inside the plant leaves. Okay, inside the chloroplasts, inside the thylakoid. Here we go. So we're going, going microscopically on this subject. Okay. Okay. Now looking at this and how that happens during the photosynthesis, you can see how light is going all the way, hitting those uh, thylakoids. Okay, and specifically hitting what the pigments. Okay, these pigments we call what? Do you remember? Chlorophylls. Hopefully this is making sense a bit. Okay, so we said that green color is the color of leaves, and there is a reason why. Because chlorophyll actually is green, that pigment, and the reason why it's green is because the um, green color is actually transmitted while the other colors are absorbed. So you have the red, yellow, purple, blue, and so on. All of these are absorbed, and what's reflected is what you see with your own eyes, which is the green color. So that these colors that are absorbed, okay, they are the ones that basically have energy, if you will, in their photons. Okay, so right, so this is talking about the whole thing here we just talked about, right? Um, okay, so wavelengths that are not absorbed are reflected, right? In this case, would be the green. Okay. Okay, you can see this on your own. Okay, so this is talking about like the how they can see all the um, all that process in front of them, right? The scientists they use this spectrophotometer, right, to be able to see the splitting of of those colors and how different colors are absorbed while others are reflected. Okay, and this is the process of that. Okay. All right. So there are three types of pigments in chloroplasts. Okay. So three types of pigments that are basically absorbing things. And these are called chlorophylls mainly, right? We said chlorophylls are those that are green inside. So we have chlorophyll A, which is the main one. So that would be the key light capturing pigment. And we will see where that's located and how important it is. Okay. Now chlorophyll B, we call this an accessory pigment. So it helps chlorophyll A with absorbing different colors and different wavelength. Okay. Uh, Carotenoids. I don't know if you've heard of that before, and we have it here in orange, looking like carrots. Okay. Why, you might ask? Because carotenoids are what basically helps our eyes, switch between like darker and lighter color faster, and it protects from extreme lights, okay, on the external environment. So these are mainly available in carrots, okay, and specifically vitamin A is what makes those carotenoids. Okay, so these are also accessory pigments. So chlorophyll B and carotenoids are accessory pigments, while chlorophyll A is the main light capturing pigment. Okay. So looking at these graphs over here, right, the absorption spectrum of chlorophyll. Okay, so we're just looking at how the different chlorophylls between A, B, and carotenoids that we're talking about, how did each one like absorb different and reflect different, uh, colors of white, of the white color actually. So So looking at the chlorophyll A, right, we can see that, so this is the y-axis will be absorption of light by chloroplast pigments, okay? We can see that chlorophyll A absorbs mostly the like purple and blue type color, okay? Um, while it also absorbs the red color and so on. Like there's a lot of different colors that it absorbs. However, when we get to the green portion, we can see that there's no absorption, and because of that, the green color is reflected. Hopefully, this is like making sense a bit. Okay. The action spectrum profiles the relative effectiveness of different wavelengths of radiation in driving a process. Okay. This is exactly what we're talking about here. So you can see when we get to the green portion, we have a less rate of photosynthesis at that, because the green is not really absorbed, it's just reflected. So the other colors are what's driving the photosynthesis. Okay, their absorption. Okay, so the action spectrum for photosynthesis is broader than the absorption spectrum of chlorophyll. Okay, so this is because accessory pigments like chlorophyll B and carotenoids, they broaden the spectrum used for photosynthesis. So a lot of different wavelengths and colors are actually absorbed from the sunlight, and that drives photosynthesis more efficiently. Okay, so the difference in the absorption spectrum between chlorophyll A and B is due to slight structural difference between the pigment molecules. So there are, I'm not gonna focus on this too much. There are differences between those two chlorophylls, and it allows them to basically absorb different wavelengths more efficiently. Okay. So in the last decade, two other forms of chlorophyll were discovered, right? There's chlorophyll D and F, and scientists have seen that these absorb also higher wavelength of light. These are, mainly used by prokaryotes, like, certain types of bacterias. Okay. They use them even in shaded conditions. Okay. Alright, so other accessory pigments called carotenoids, we talked about this, right, are yellow or orange, right, because they absorb violet and blue-green light. Okay, so again, it's always the opposite. So if violet and green light are absorbed, then you would expect, if we have violet and green is on this side, and red and orange is on this side, so if they absorb the violet, then you're gonna basically reflect the opposite. Exactly when we talked about the chlorophyll A, which basically absorbs all the other colors and reflects green. Okay. So they broaden the spectrum of photosynthesis. As we said, it's an accessory pigment, and they're photoprotective too. Remember. Same as in our eyes, okay? So it protects from, the external environment. Okay, so let's talk about the pigment chlorophyll specifically. We're talking about chlorophyll, just different pigments, and mainly at the end, chlorophyll A pigment, okay? So when a pigment absorbs light, so you have the light hitting those pigments, okay, as you can see here. Okay, this green circle is a pigment, it's chlorophyll, right? Chlorophyll is a pigment. The electrons inside that pigment is excited, and if you remember from like the beginning 2 chapters, right, when we talked about electrons and protons, when we say electrons are excited, that means electrons have basically went from ground state, or from a shell that's around the nucleus, or the nucleus of the atom, to a shell. It goes to a higher shell further away from the atom. Okay, so we call this you know, exciting the electron. Okay, so it becomes unstable in this situation. Okay, so it goes away from the uh, the nucleus, okay, to a higher state. It gets excited, and because it's unstable, it goes back to the ground state. Okay, so when it does that, okay, we have basically heat, we have and photon that are basically products of that, or results of that. Okay, so this is what we want. We're trying to create the energy to be able to finally excite the main chlorophyll A at the end. We will see you know, how that happens in a bit, the whole thing. I'll show you a summary of that. Okay, so in isolation, some pigments also emit light, an afterglow called fluorescence. This is just an example of that. Okay, this if we had put like um, pigments in an external environment, not inside the chloroplast, then you know, you're able to see like what's being absorbed and what's being emitted. Okay. Okay. Now this is new, right? We're gonna just dive into it little by little. Okay, so this is the light reaction. This is what happened inside a light reaction. Okay. Um, And in the light reaction, the main system that basically starts the whole process is the photosystem, we call it. Inside that photosystem, as you can see here, so the whole thing, this purple structure, is actually called a photosystem. Okay? Okay, so inside that, we have a light harvesting complexes. That would be just this purple. Okay? So the whole thing is the photosystem. The purple is just light harvesting complexes. Okay, in the middle is where you have your reaction center complex. Okay. You can see those green circles, and these are the pigment molecules we were talking about. Okay? So, you know, it could be accessory pigments like chlorophyll B, carotenoids. Okay? However, in the middle here is your main pigment that captures the light and um, basically excites its electron as a result at the end. Okay, this is where we call what we call the chlorophyll A molecule. See, it's just a pair. That's normally they're always a pair like that. That's how you find them inside a thylakoid. So this is basically the membrane of a thylakoid. On the outside of that membrane is the stroma. Remember that? I'm just letting you know where we are. Okay. Okay. So on this side is your thylakoid space, because this is the thylakoid membrane. So inside here is the thylakoid space. Okay. So reaction center complex. This thing inside here is in an association of protein, right, holding a special pair of chlorophyll A, as we said. Okay, and that will be a primary electron acceptor, which is this one. Okay, so just to give you a little summary, so we have light energy, right, in the form of photons from the sun. This light energy basically hits that pigment, okay, it's you know, accessory pigments, and basically electron inside this gets excited. Okay, so, but one thing you want to know is that this arrow does not actually represent transfer of electron yet. This arrow just represents transfer of energy. So each one of these pigment, when it gets excited because of that light energy, it transfers that energy over to the next one as you can see here. So it's like one after the other, until it reaches finally your pair of chlorophyll A molecules, as you can see here. And that would be basically the main one that its electrons actually are excited and actually is transferred as a result. Here would be where your primary electron acceptor is. Okay, what would be the primary electron acceptor in this situation? It would be the NADP plus, right? This is where the electron gets attached to, and we have an ADPH. Okay? Okay. Okay, so we just talked about all of this, how the energy is transferred to the chlorophyll A molecule. Okay. And then these are special because they can transfer an excited electron. So these outside ones, they only transfer the energy. This one actually transfers the electron over to this primary electron acceptor. Okay, so primary electron acceptor, right, in the reaction center accepts those excited electrons, right, and is reduced as a result. We said it's reduced to what? An ADPH, okay? Solar-powered transfer of an electron from chlorophyll A molecules to the primary electron acceptor is the first step of light reaction. So this is your first step. There are more steps because why? We have to produce what else in the light reaction? So we're producing NADPH and ATP, right? To be able to have enough energy for Calvin cycle to take place. Alright, so there are two types of photosystems in the thylakoid membrane. Okay, so we were talking about one system here. That would be the photosystem 2. Okay? The photosystem is said to be functioning first before photosystem 1. So these are basically embedded in the thylakoid molecule. Both photosystems. They work together. You will see how that happens. Okay, so in photosystem 2 we have we call this chlorophyll A pair, we call them P6AD because these are chlorophyll A that are specialized in absorbing wavelength that are of 680 nanometer long. Okay, so I'm gonna just go back in a bit just to show you so you're not confused what the heck is this P6ADP is. Okay, so let's go ahead and look at this light spectrum a bit and just show you where we are. Okay, 680 is around here. You see that? See how this is being absorbed? Okay, even looking at the full spectrum here, we are here. Okay, and we will see that the next one is actually in the 700. Let me go back. Sorry for doing that, but I just wanted to show you where we are. So 680, okay, it's absorbing towards the red colors. Okay, so photosystem 1 is best at absorbing a wavelength of 700. So they're next to each other, but they're different. Okay. So let's talk about this electron flow, and we call this linear in this situation. In plants, we call this linear electron flow. So during the light reactions, there are two possible routes for electron flow. We have cyclic and linear. Linear electron flow, the primary pathway, right, involves photosystems, 2 in 1, and produces ATP and ADPH at the end using light energy. So hopefully you know this by now. Okay, so now we're going to talk about the actual steps of photosystem, or the photoreactions, okay? And specifically the linear electron flow. So how does actually electrons gets transferred all the way until the end? So a photon hits a pigment as we said before, right, in the light harvesting complex of photosystem 2 first. So this photosystem 2 comes first before photosystem 1. Okay, so now when this gets excited, the energy gets transferred, okay, among pigment molecules, as you can see here, until it excites P680, which is a pair of what? Chlorophyll A, right? Pigments. An excited electron from P680 is transferred, as you can see here. This is why this is special, because it can transfer actually an electron, not just the energy, okay, to the primary electron acceptor. Okay, now when this happens, we call this P680 plus. I know this is getting a bit confusing, but bear with me. So you lost an electron here because of excitation. What happens when you lose an electron? These becomes oxidized. It's exactly like NAD plus or all of those molecules we were talking about. So you lost an electron here, you now have a P680 plus. We will see why this is important in a bit. Okay, now just stay with me in a bit here. So H2O, which is water, is when this actually comes in. So water comes in while also light hits and do its own thing, okay? Now, do you remember from the beginning we said that electrons are actually coming from what? From water itself, okay? From the splitting of H2O, this is how we have the main water, okay? The main electrons, I'm sorry, okay? And this is how we release this byproduct O2. Okay, so an enzyme catalyzes the split of H2O into 2 electrons, 2 hydrogen ions, and an oxygen atom. So we have an enzyme that splits this water into 2 protons or 2 hydrogen ions, as you can see here, 2 electrons, as you can see here, and the O2 oxygen. Now it's half O2 here because, you know, another splitting of H2O molecule next to it gets you another half of the oxygen. They combine together and gives you this O2 gas molecule that gets released out of the pores. Okay. So let's continue with this. The electrons are transferred to the P680 plus. So do you remember that? We created P680 plus here. Okay, you remember that? So when we have an oxidized pigments here, that means they're able to accept electrons now. So basically follow this with me. So number one, this happens, the light gets basically do its own thing. Number two, the electron is excited from P680 and it gets basically captured by this primary electron acceptor. Okay, now when this happens, P680 becomes P680 plus or oxidized. Now it's ready to capture electrons from the water. Okay, so this is how this whole thing happens. Okay, and so we replenish those electrons here and the whole thing happens again. It's a recycling. Light actually goes again and excites those electrons that are here again, and they get released and so on. It's like a little machine, okay? So now we talked about O2 will be byproducts, right? They're released in the air, okay? We said that we use those electrons from the splitting of water. What about those hydrogens? Do they just get lost somewhere? No, we're actually also using them. They get released in the thylakoid space. So this is actually in the thylakoid membrane happening. Now H plus gets released in the space of thylakoid. So it's inside the thylakoid now. Not outside. Outside would be the stroma. Okay? They're inside the thylakoid space, which we said the thylakoid is a disc, right? So inside that would be the space. The oxygen atom combines with another oxygen atom, okay, as I mentioned before, from splitting of another water molecule, and they form, as a result, the O2 gas. Alright, now we're going to talk about how these electrons actually gets going even further. Okay, so each electron falls down an electron transport chain. Here you go. So we have that electron transport chain exactly like what happened in the mitochondria. In cellular respiration, same thing. Okay, so that electrons gets transported into PQ, okay, then the cytochrome complex, then into PC. Okay, so you don't have to remember this, but it's PQ is plastoquinone and PC is plastocyanin. Okay, it's just getting transferred between them. Okay, and let's see here. So let me see. I don't want to lose this thought. So each electron falls down the electron transport chain from the primary electron acceptor right here, right, of photosystem 2. Now we haven't even talked about photosystem 1 yet. So it's going to fall down from here all the way until PS1, which is photosystem 1. Okay, energy released by the fold drives the creation of proton gradient across the thylakoid membrane. Okay, so when this is basically fallen, right, the electron transport chain includes the, okay, we said all of this stuff, right? Now this is where the H pluses that were released from here come in, right? Energy released by electron transfer is used to pump H plus into the thylakoid space. So the electrons is flowing down, into the electron transport chain, and as a result, they basically they create those this mechanism, and where H plus gets pumped, okay, across the thylakoid membrane. Okay, so we call this proton gradient. Okay, when H plus gets pumped across the thylakoid membrane, we have also the same process that happens in cellular respiration, and this is where chemiosmosis happens. Chemiosmosis is exactly the same thing. You have ATP synthase, using basically those hydrogen atoms that are concentrated now. When they basically fall through the ATP synthase, that's when we start creating ATP here. Okay. Okay. So this is chemiosmosis, and how ATP is produced. Remember, in the light reactions, two things are mainly produced: ATP and ADPH. Okay, this is the final electron acceptor. All right, let's talk about now photosystem 1, which is similar to photosystem 2, but a bit different. Okay, so let's talk about it. So light also hits that system as well, as you can see, same way it hits that photosystem 2. Okay, when that happens, these pigments get excited, same way, and they release energy. Now, these pair of chlorophyll A here, we call them P700 instead, so they're specialized in absorbing P700 wavelength lights. Same thing, this chlorophyll A pair, actually when it gets excited, it actually releases electron, okay, and this electron gets accepted by this primary electron acceptor. What happened here? That P700 becomes positive. Again, when that electron gets released, same way. So we say P700 plus is oxidized. Okay, now what happened here is that when it's oxidized, it accepts the electron passed down. Here we go. Hopefully you can see how this is all coming together. So these electrons that are being transferred from photosystem 2 down the electron transport chain, they now can be actually attached to P700 and accepted by P700 because it's oxidized. When that happens, P700 becomes reduced because it accepted those electrons. Okay? Now, the same thing happens again. Now P700 is reduced, so it's ready for another light reaction to happen here, and then those electrons are again transferred to the primary electron acceptor again. So you can see this whole thing, how it kind of recycles itself and how efficient it is. Okay, so let's talk about, just to summarize the whole thing. So we're going to talk about each electron falls down an electron transport chain from primary electron acceptor of PS1. Okay, so we're continuing here. Okay, we have another electron transfer chain. This time we have FD complex here that basically allows this transfer to happen instead of PQ and PC and cytochrome complex. So it's shorter. Okay. FD is protein ferredoxin. So there's no protein gradient or ATP produced by this electron transport chain. So it's shorter. Therefore, there's no ATP being produced like you see here between photosystem 2 and photosystem 1. So there is no chemiosmosis here. Okay. Now, the final electron acceptor that accepts the whole thing, how the electrons are being transferred, this is what we need mainly for what? For the Kelvin cycle to take place. So this NADP plus is actually already here, located, and using an NADP plus reductase, that's an enzyme, okay, it reduces NADP plus using those electrons to NADPH. Okay, we need 2 electrons to reduce NAD plus to NADPH instead of 1. Okay, so the electrons of NADPH are a higher energy level than they were in H2O, obviously, right? So it's exactly the opposite of cellular respiration. So when cellular respiration we had like higher energy, and then we ended up with lower energy molecule. Here we had a lower energy molecule, and we're ending up with a higher energy molecule. You can see the differences between endergonic and exergonic reactions. Okay, so the formation of NADPH also removes an H plus from the stroma. Okay, as you can see here. Alright, so this is basically a summary of the light reactions. Okay, and this is just a depiction of the whole thing, how that happens. You can look at it yourself, okay? Let's see here. Okay, so we talked about the linear electron flow earlier. Now we're going to talk a bit about the cyclic electron flow. So in cyclic electron flow, photoexcited electrons cycle back from FD, okay, to the cytochrome complex instead of being transferred to NAD+. So look at photosystem 1, where the FD is the main thing going on in the electron transport chain. So instead of basically going further and those electrons gets accepted by an NADP+, what they do is they recycle in between the photosystem 2 and photosystem 1. They go back to the cytochrome complex and PC, and ATP is created as a result, obviously, because this electron transport chain is capable of producing ATP. Okay, and then basically it gets accepted by the primary acceptor here in photosystem 1, and then does this again and again and again. Okay, so yeah, electrons are passed to P700. Here you go, right here in photosystem 1. Chlorophyll and PS1 reaction center via the plastocyanin molecule. Okay, so it goes from PC to P700 chlorophyll. And then cyclic electron flow uses only photosystem 1. Okay, so you can see that it's just using this and it recycles back. It produces ATP but no NADPH. Obviously it's not going further. Or oxygen results from this process. Okay. These cyclic, electron flow, they're actually being used by photosynthetic bacteria. So they only have a single photosystem related to either PS2 or PS1. So they're able to produce ATP to maintain themselves, but they're not able to, do what plants do, which is go further and do the Calvin cycle because of their ability to reduce NADPH. Okay, so they're able to just do ATP alone to maintain themselves. Okay, so they generate ATP during photosynthesis only. We're talking about just these photosynthetic bacteria only. Okay, we're not talking about plants here. Alright, photosynthesis may have first evolved. This is talking about just a little bit of evolution, right? And it's ancestors of these bacteria and form similar to cyclic electron flow. Okay, cyclic electron flow is probably part of an evolutionary leftover in organisms with both photosystems. Okay, cyclic electrons flow may have photoprotective capability, right? Plants that do not have it grow well in low light, but cannot grow well in intense light. It's just protective capabilities that this cyclic gives them. Okay, so this is just a comparison between the chemiosmosis of chloroplasts and mitochondria. Remember, like we saw similar pumping of H pluses ions that create ATP at the end. Okay, so they both generate ATP. Okay, the electron transport chains pump protons, right, through what the ATP synthase. Okay, and that basically allows the phosphorylation of ADP by putting another phosphate on there and creating ATP, which is the energy molecule we want. Okay, and this is showing you that chemiosmosis, how they're similar and how they're different. So in mitochondria, right, protons are pumped to the intermediate space and drive ATP synthesis, as we know by now, right? So same thing, it's happening here. This is just showing you like a little difference between, the mitochondrial structure on this side, chloroplast structure on this side. Okay, you can see how similar they are. This would be your the membrane of both organelles. Okay, you can see how the electron transport chain here allows the transfer of H plus to create that gradient on the outside, and then this ATP synthase allows this when the H falls down their concentration gradient. It allows this ATP synthase to actually start working and producing ATP by phosphorylation as a result. Okay. In mitochondria, protons are pumped to the intermediate space, as you can see here, right here actually, and drive ATP synthase as they fuse back into the mitochondrial matrix. That's the matrix. Okay. In chloroplasts, protons are pumped into the thylakoid space, same thing, and drive ATP synthesis on the stroma side, right, of the membrane as they diffuse back into the stroma. Okay? Alright, so ATP and ADPH are produced on the side facing the stroma, where the Calvin cycle takes place. The Calvin cycle is in the stroma. Okay? Alright, so this looks complex, but this is what we were just talking about the whole time. So here you go. Photosystem 2 here, photosystem 1 here. You have your electrons depicted here being flowing here with this yellow arrow. See that? You have light hitting both photosystems, as we saw earlier. Okay? We have, as a result, these protons, right, H pluses, are being formed from the splitting of water molecules. You see that? Okay. and we have a higher H plus concentration in this case inside the thylakoid space. And when we have this higher H plus concentration, these H pluses are going to fall down their concentration gradient um, into this ATP synthase, right, and create ATP as a result using phosphorylation. You see that? This is what we just talked about. Okay. Now, looking back on the photosystems 1 and 2, right, we can just follow the electron path, okay, and how this final electron acceptor molecule, which is called NADP plus, okay, is reduced using NADP plus reductase enzyme. This is an enzyme, excuse me, and then it reduces it to NADPH. Both NADPH and ATP are basically needed for the Calvin cycle to take place. Okay, you haven't seen any carbon dioxide here yet. Carbon dioxide will come in here in a bit. Okay. Do you see that? This is exactly where we are. Hopefully you're following the process of thinking here, okay? Okay, let's talk about the Calvin cycle now a little bit. So Calvin cycle uses the chemical energy, right, of ATP and NADPH that was harvested in the light reactions we were talking about to reduce CO2 to sugar. Okay, so we're creating sugar, which is a more complex molecule from CO2. The Calvin cycle, like the citric acid cycle in cell respiration, regenerates its starting material after molecules enter and leave the cell. Okay, so so the Calvin cycle basically is anabolic, right? It's creating, a more bigger complex molecule sugar than CO2. That's why it's anabolic. So we're building a bigger molecule in comparison to cellular respiration, where we broke down glucose and we made it into like very small molecule, which is carbon dioxide. Do you remember that from the citric acid cycle? So let's see here. So yeah, the Calvin cycle is anabolic. It builds sugar from smaller molecules, right, by using ATP and the reducing power of electrons carried by NADPH. So you can see how Calvin cycle uses those ATP and ADPH produced from the previous photoreactions, light reactions, to be able to do what it needs to do. Okay. So the carbon enters the cycle as CO2 first, and leaves as a sugar named glyceraldehyde 3-phosphate, so G3P as a abbreviation. Okay? So for net synthesis of one G3P, so if we want to create one molecule of G3P, the cycle must take place 3 times. So it's a cycle, right? The Calvin cycle, kind of like citric acid cycle. So, but it has to do 3 times the cycle to be able to do the G3P. So the G3P has 3 carbon inside them. So because of that, it needs to do it 3 times by using 3 molecules of CO2. So it can fix carbon from each CO2. You see, like this has 1 carbon, another has another carbon, and a third one has another carbon. So the Calvin cycle has 3 phases. It's got the carbon fixation first, which is where this takes place. Then it's catalyzed by Rubisco. So this is also another enzyme that it's also, there's a molecule also called Rubisco in there. But this is an enzyme that allows the carbon to be fixed together to be able to create just G3P in a bit. Okay, we will talk about it in a bit. Then CO2 reduction is your second phase. Then regeneration of the CO2 acceptor RUBP happens in this third phase. So let's talk about each phase in a bit. Okay, so you have basically your input of 3 CO2s, as we said. So we need 3 CO2s to create one of our G3P molecule, as we mentioned here. So we got 3 carbons are coming here. We have Rubisco, which is the enzyme, okay, that fixes those carbons. It fixes those carbons to this RUBP, or we call it ribulose biphosphate. Okay, you see that? It's a 5-carbon chain molecule. It's basically a carbohydrate already. Okay, so it's already available in the stroma. Okay, so the binding of CO2 to the 5-carbon sugar, this RUBP, as you can see here, using Rubisco. Okay. Rubisco RUBP is basically the carboxylase oxygenate, however you pronounce that. Okay, so but we're just gonna say Rubisco. That's what everybody calls it in biology. Okay, so we're just gonna say Rubisco. Okay, so that's an enzyme that does that. All right, now this, as a result, creates a 6-carbon intermediate molecule, and it's split. It's immediately split into into 2 molecules of 3 phosphoglycerate. Okay, for each CO2 that's fixed. Okay, so let's look at it this way. So you have basically the addition of carbon dioxide to this creates a 6. You see, you can even count them, right? So 1, 2, 3, 4, 5, 6 carbon intermediate, as you can see here. Then this basically gets split into 2 of these 3 phosphoglycerate molecules. So each one has 3 carbons now instead of 6. Okay? Now, let's continue. Phase 2, so this was what? Phase 1. We said that phase 1 involves carbon fixation. Okay, see how it's catalyzed by Rubisco, and we talked about this here. Now phase 2 is talking about CO2 reduction. So let's talk about this right now. So each molecule of the 3 phosphoglycerate that we create, so we created 2 of these 3 phosphoglycerate molecules, is altered through phosphorylation by 6 ATP and reduction by 6 NADPH to ultimately produce the G3P sugar. Okay, so just stay with me in a bit here. So you might be asking, like, where did this ATP come from and where did this NADPH come from? These came from the light reactions that we were talking about. So this is the step that actually Calvin cycle uses these harvested energy and harvested electrons to create this G3P sugar. Okay? Again, this is where the ATP and NADH are being used. So they use 6 of them, okay, to do basically each molecule of 3 phosphoglycerate. Each molecule of phosphoglycerate is basically reduced. Okay. As a result, if this is reduced, okay, then we say that NADPH is what is oxidized. This is your oxidized form. Okay. Hopefully this is making sense. So this is where CO2 reduction comes to place. Do you see that? So this molecule is reduced to G3P. So for every 3 CO2 molecules that are coming from here, right, we said that we need 3 CO2 molecules to create one of this phosphoglycerate molecule. Or not phosphoglycerate, but would be the intermediate molecule, which is before the phosphoglycerate. But you create 2 of these 3 phosphoglycerate molecules for every 3 CO2 that are being basically used. Okay. So for every 3 CO2 molecules that enter the cycle, 6 molecules of G3P are formed. You can follow the numbers here. So you have 6 here that are being formed. Do you see that? Number 6. Okay. Now, only one of these can be counted as a net gain of carbohydrate. Okay. So remember, the process of photosynthesis on this side, we get what? We get a molecule of sugar or carbohydrate. Okay? This is where this comes from. So you get one G3P that the plant uses to build a glucose out of. So G3P is not the actual final product, right? G3P is in like a, you know, a reactant that, or a small portion, right, that the plant uses to build glucose from and other organic compounds. Okay. Now we had 6 here, we said, right? 6 of these G3Ps that have been harvested through the carbon fixation. One of them are used to build glucose. Now you might say, okay, what about the other 5 out of the 6? Okay, so the other 5 actually continues the cycle to produce RUBP again. That would be your phase 3. So regeneration of the CO2 acceptor RUBP. Do you see it? That would be your third phase. That requires 5 molecules of G3P for this to happen. Okay, let's talk about it. So the remaining 5 molecules of G3Ps, you can see here 5 of them. Okay, they are rearranged in a complex series of reaction yielding 3 molecules of RUBP. Okay, 5 of these are rearranged using ATP again. More ATPs are being used here. So you can see how all the energy that we harvested from light energy, and in the light reactions, we used all of these here to help create the sugar and also help recycle the material again, which would be, in this case, what? RUBP. Okay, if you don't have RUBP, then you cannot fix carbon dioxide to it and create everything that you want. Okay? Okay, so 3 additional molecules of ATP are used to facilitate the regeneration of RUBP. So 3 are used here, 6 are used here. So if I ask you what's the total how many ATPs are used per cycle of the Calvin cycle, it would be what? 9 ATPs and 6 NADPHs. Okay? I know this is new stuff, I know this is difficult, but just keep at it and keep repeating it. Watch those videos so you can understand this stuff, and pay attention to my teaching a little bit, and you will get the hang of it eventually. Okay? It will just take some time for you to understand it. Alright, so this is a summary of the whole thing. The whole photosynthesis system. Do you remember all of this now? Okay. This ATP and NADPH are used in this Calvin cycle. Okay? When we use them, we actually create an oxidized NADP plus and ADP. Here we go. So ADP here, okay, and NAD plus here. You see them? Okay, we need those to be able to recycle the whole thing. Okay. As a result of the Calvin cycle, we have sugar in the form of sucrose. So after we create that G3P molecule out of the 6, we have one of them that gets used, okay? When we create that one, and we do it several times, right? We are able to put together, okay, sucrose that the plant uses. Okay, we can also create starch for storage. If the plant wants to store its carbohydrates for later on, it can do so as well. Okay, so here are your reactants on the top. Here are your products on the bottom of photosynthesis. Alright, you can see this on your own. So for the net synthesis of one G3P molecule, so to create one G3P molecule, as we said, the Calvin cycle consumes 9 molecules of ATP and 6 molecules of NADPH. We said this here, right? 9, so 6 plus 3, and 6 NADPH. Okay? The light reactions regenerate the ATP and NADPH. Okay? We know this by now. Okay? We need these to be regenerated so that more G3P molecules can be produced and life goes on. Okay? G3P is the starting molecule for metabolic pathways, or anabolic pathways specifically, to synthesize those organic molecules like sucrose that the plant uses, or starch, okay, that the plant also stores. Okay, now, what happens if you have, not that much water to be able to harvest the necessary electrons? Okay, this is where dehydration. When we have less water, we have dehydration, and it's a major challenge for terrestrial life for plants. Okay. So how do actually plants cope with this? Okay. So plants have metabolic adaptations, so they adapt to help conserve water. So whatever they were able to absorb before dehydration happened, they try to conserve it. We will see how that happens. Okay. Uh, These adaptations, however, often involve trade-offs, unfortunately. Okay? So one important trade-off is the balance between photosynthesis and water conservation. So plants want to do photosynthesis as part of their, part of their life cycle. That's what they do. However, they also want to conserve water when water is scarce, whatever that word is. Okay? So when the plants, when they're dehydrated, they want to conserve their water. Okay? So on hot, dry days, when there's not a lot of water, plants close stomata. You might be asking, what's stomata? Stomata are those pores that we talked about earlier in the leaves that allow O2 and CO2 and water to come in and out. Okay? So when they close the stomatas, those pores, they conserve H2O in the process. Okay? So H2O that have been already absorbed gets conserved. However, this limits photosynthesis. Why? Because closing the stomata reduces basically excess CO2. So from photosynthesis, right, we need what? CO2 to proceed with photosynthesis. So it reduces access to CO2, so the CO2 from the outside to the inside of the leaves drops, right? What about the O2 that are being produced as a result from the photosynthesis, right? That would be a product of photosynthesis, as we said. This increases inside because it's not able to get out of the leaves, okay? These conditions favor an apparently wasteful process called photorespiration. Okay, so it's a process to just maintain themselves. Okay, they photorespire. We will see what happens here. Okay, so in photorespiration, we have different types of plants. We have C3 plants, we call. Okay, in which the initial fixation of CO2 via rubisco, do you remember that, in the Calvin cycle, forms a 3-carbon compound, 3-phosphoglycerate. This is the normal process, right? So C3 plants are your normal plants. Okay, so this is the normal photosynthetic pathway. So in phase 1, we get this 3-phosphoglycerate. Do you see that? Okay, let me just continue. I'm just going back to show you where we are, okay, to keep you organized in your thinking, okay? So it forms this 3-phosphoglycerate using this C3, inside the C3 plants. Okay, so in photorespiration, okay, when you have like dehydration and so on, Rubisco actually binds with O2 instead. So remember, we said that when those stomata pores gets closed up, we have a less amount of CO2 going inside the leaves and more amount of O2 instead. So Rubisco, instead of binding with CO2, it binds with O2 in the Calvin cycle. And when that happens, actually 2-carbon compound is produced instead of the 3-carbon compound you see here. Okay, so photorespiration is costly because it consumes O2 instead, and organic fuel without producing any ATP or sugar. So if we can't use CO2 to produce this 3-phosphoglycerate compound, we won't be able to create the sugar at the end that we said. Okay, and we won't be able to produce ATP as a result either. Okay, ATP is produced from what? From the sugar when the plant actually uses whatever it made, sucrose in this case, to consume it. Remember, plants have mitochondria too, right? So it consumes this carbohydrate and produces ATP. So when this whole process doesn't work together, you don't have enough ATP being produced or sugar. So it is thought to be an evolutionary relay because Rubisco first evolved at a time when the atmosphere had far less O2 and more CO2. So you can see that Rubisco actually is able to use O2 instead of CO2 in this situation. Photorespiration may provide some protection from damaging products of the light reactions that build up when the Calvin cycle slows due to low CO2. So if we have low CO2, which what the Calvin cycle uses, right? And you have more production of what? Because of light reactions. So the light reactions are still happening, yeah, right? They have nothing to do with CO2. So light reactions from the sun are happening. So as a result, you have a buildup of NADPH, right? And you have a buildup of ATP as well inside this. I know I said ATP here, but this ATP in photosynthesis is different than the ATP that plants actually consumes to stay alive. Okay? Now, okay, so in photorespiration, Rubisco adds O2 instead of CO2 in the Calvin cycle. Okay, we said this, right? Photorespiration consumes O2 and organic fuel and releases CO2 without producing ATP or sugar. Okay, this is just repetition. In many plants, photorespiration drains away as much as 50% of the carbon fixed by the Calvin cycle. You can see the process is now going like against the plants here. Okay, in some plant species, alternate modes of carbon fixation have evolved to minimize photorespiration and optimize the Calvin cycle. These would be different than the C3 plants. These would be C4 plants and so on. Okay, C4 plants, as you can see here, that's an anatomy of a C4 plant. These are the examples like sugarcane and corn plants. So they're not the green leafy plants. Okay, these would be C3. The sugarcane and corn would be C4 plants. Remember that. Okay. Looking at these C4 leaf anatomy, we can see their anatomy is composed of what? Mesophyll cells that are on the outside. Then attached to them on the inside going in is a bundle sheath cells, as you can see here. And in the middle we have these vein or the vascular tissue which transport all the nutrients. Okay, so in the case of C4 plants, let's talk about them, how they're different than C3. So they minimize the cost of photorespiration, okay, by incorporating CO2 into a 4-carbon compound as the first product of the Calvin cycle. So they use that CO2 and they incorporating into 4 carbon compound, okay, instead of your normal, you know, 5 carbon compound, which is RUBP, okay. So they incorporate C2 into 4 carbon compound, and that would be their first product of Calvin cycle, okay. C4 has evolved several times and is used by several thousand species. Like, there's a lot of them, right? And mostly what we know is the sugarcane and corn as well. So what happened is that in hot, dry weather, C4 plants partially close, so not fully, but partially close their stomata, conserving water but reducing CO2. So they're not completely preventing CO2 from coming in, right? They're partially reducing it, okay? And they conserve water when they do that as well. Photosynthesis begins in mesophyll cells, okay? Mesophyll cells, we said, have what? Chloroplasts inside them, right? So they have like tens and tens of chlorophyll organelles inside them, okay? So photosynthesis starts there, right? But it's completed in the bundle sheath cells. So it starts here, but then it gets completed in the bundle sheath cells instead. Okay, so looking at this right now, just to give you, an idea of what's going on. I know this is also complex, but let's just go through it little by little. So let's talk about sugar production in C4 plants. It basically occurs in 3-step process, as you can see here. So this is a cross section of what you're seeing here. Okay, so this would be your your mesophyll cell. This would be your bundle sheath cell. Okay, it even tells you here. Okay, and this would be your vascular tissue. Okay, so the production of the 4 carbon precursors is catalyzed by the enzyme PEP carboxylase in this case in the mesophyll cells. Okay, so here's oxaloacetate, okay, in this cycle, and we can see how CO2 is actually used to fix it to the oxaloacetate using the PEP carboxylase enzyme. Okay, so it's different than the Calvin cycle. Okay, the Calvin cycle uses CO2 to fix it onto the RUBP, carbohydrate molecule. Okay. Now, PEP carboxylase has a higher affinity for CO2, right, than Rubisco does. So this is a comparison of what happened in C3 and C4. Okay, C3 is where we have the normal Calvin cycle. C4, we have PEP carboxylase working instead of your Rubisco enzyme. Okay, so it can fix CO2 even when CO2 concentration are low. So because it has a higher affinity to CO2, it does not need to have a higher concentration of CO2 for it to fix CO2. Okay, so it's very, like, you could say, like, highly sensitive to CO2. Okay. Now, these 4 carbon compounds are exported to bundle sheath cells, as you can see here. So they're exported here. Inside the bundle sheath cells, okay, they release CO2. So CO2 starts from here, gets released in the bundle sheath cells here, and that CO2 is now used in the Calvin cycle. Do you see that? The Calvin cycle uses what? The Rubisco. Okay? So you can see how this is a kind of an efficient way to use a lower amount of CO2 to actually be able to produce enough quantities to be able to finally be used in the Calvin cycle and produces your sugar at the end, and let the plant, like, you know, maintain itself. Okay, so pyruvate is transported to the mesophyll cells, okay, where one ATP is used to convert it back to PEP. Okay, so from malate we have a production of CO2 and pyruvate. Pyruvate is a 3-carbon molecule. Hopefully you know this from last chapter's cellular respiration. So this actually recycles that 4-carbon carbohydrate and gets recycled to the mesophyll cell, but it needs basically ATP to be recycled. So you can see that actually energy is being used. Okay, the process costs the plant energy to do so. So this ATP is generated using the cyclic electron flow. You remember in the cyclic electron flow, we have ATP being produced, right? Instead of the linear. Okay. Since the Industrial Revolution in the 1800s, right, we know that CO2 levels have risen greatly. Increasing levels of CO2 may affect C3 and C4 plants differently, perhaps changing the relative abundance of these species. Okay. The effects of such changes are unpredictable and cause for concern. Okay, so suitable agricultural land is increasing due to the effects of climate change, while the world demand for food continues to increase, right? C4 photosynthesis uses less water and resources than C3 photosynthesis. Okay, because C3 needs more carbon dioxide to be able to continue. Okay. Scientists have genetically modified rice, which is normally a C3 plant, to carry out C4 photosynthesis. So C4 is basically more efficient, if you want to call it this way. Okay? And you can see why. This process makes it more efficient here. Okay? Due to genetically modifying C3 rice into C4, they predict that they will have 30 to 50% increase in yield compared to C3 rice. Now, let's talk about cam plants. This is a third type of plants. When we talk about cam plants, we're talking about, you know, plants that are living in the deserts, basically, like cactus, like I guess would be pineapple. I can't think of any more right now, but these are two examples. Okay, so in the deserts, you can imagine that there aren't enough water. Okay, and the actual environment is different. So these plants open actually their stomata only at night, incorporating CO2 into organic acids. Okay, so they open them at night, these pores. They get that CO2 from the outside only at night. They convert that CO2 into organic acids, okay, and they store them in the vacuoles. Okay, now during the day, these stomata closes. And when they close, what happens? CO2 is released from organic acids. So this is the way that these cam plants are actually conserving CO2. So they capture it at night. They use it actually in the day. Okay? They use it to what? To go on and produce sucrose, using the Calvin cycle, right? As we normally learn from how CO2 is being used in the Calvin cycle. So CAM pathway is actually similar to C4 pathway in that they both incorporate CO2 into organic intermediates. Okay, so again, CO2 is incorporated into organic acids, which are, same thing like intermediates, like what we saw here. Do you see that? Okay. Before it enters the Calvin cycle. Okay, so that's a way for the plants to store and use the CO2 more efficiently. Okay. C4 pathway structurally separates the initial steps of carbon fixation from the carbon cycle. Yeah, they're structurally different. Why? Because this first step happens in the mesophyll cell, while the second step happen in a different cell. It's a bundle sheath cell. However, in the CAN pathway, these steps all occur in the same cell instead. Okay, but they're separated in time, right? As we said, at night, these carbon dioxides is basically converted to those um, organic acids, and in the day, this carbon dioxide gets incorporated into the Calvin cycle. And here's exactly what we're talking about. Okay, here we go. So this is one cell, and this is two separate cells. This is a C4 sugarcane. This is a pineapple cam plant. Okay, you guys can see all of this. Alright, so photosynthesis is essential for life on Earth, right? That's just, we're just reviewing all of this. So the energy entering chloroplast, right, sunlight, gets stored as chemical energy in organic compounds. Hopefully we learned this by now. Sugar made in chloroplast supplies chemical energy in carbon skeletons to synthesize, organic molecules of cells. So that sugar, that's harvested from the Calvin cycle, basically gets put together in order to create your, different molecules and starch and so on. Okay? So just a summary, what goes on in a light reaction, what goes on in a Calvin cycle reaction. Light reactions are carried out by molecules in the thylakoid membranes. Remember that. If they convert light energy, right, they're called light reactions, to chemical energy of ATP and NADPH. Both of these are used in the Calvin cycle. Okay, water is also split up and release O2 to the atmosphere. This is where we get our electrons from. Okay, in the Calvin cycle reactions, it takes place in the stroma instead of the thylakoid. It uses the ATP and NADPH that was harvested from light reactions to convert CO2, right? We said how many ATPs are being used? 9. How many NADPH? 6 in the Calvin cycles reaction, okay? That produces the sugar G3P. That G3P sugar is basically incorporated with more G3Ps or to create sucrose at the end, or to create starch and other carbohydrate molecules. Then it returns ADP and NAD+, which are oxidized, back to the light reactions to be used again and carry electrons and energy again. Okay, and that should do it for this chapter. Hopefully this was clear to you. I know this is new, hard material. Just keep studying and let me know if you have any questions, and I'll see you in the next lesson.